G'day everyone, my name is Jade Russell and I'm from Cesar Australia, and I'd like to welcome you all to today's Research in Action webinar. This will be the first of two Research in Action webinars that we'll be uploading. Before I introduce you to today's presenter, I'd just like to acknowledge that I'm recording this webinar from the lands of the Wurundjeri people, and also acknowledge the traditional custodians of the various lands on which everyone is watching today. So today's presentation will have a heavy focus on the evolution of insecticide resistance in Australian blue-green aphids. Dr. Evatt Chirgwin, one of our research scientists here at Cesar Australia, has done a lot of research into this exact question. And today he's going to be going over whether blue-green aphids have evolved resistance, some chemical, biological and cultural controls, some useful aphid monitoring methods, and some really interesting resources as well, which can be found in the description below, and also how you can help us by sending in some aphid samples for resistance screening. I'll now pass the stage on to Evatt so we can tell us about the evolution of resistance in blue-green aphids.
All righty, let's get started. So when it comes to sports, Australians love to come first. And we often celebrate the many medals and trophies that come with first place. Now, today I'm going to talk about Australia coming first at something, but it's not something we necessarily want to celebrate. In fact, something we're a bit worried about. And that is the first time this worldwide crop pest, the blue-green aphid, has evolved insecticide resistance anywhere in the world, and it's happened here, Australia. Hi, my name is Evatt Chirgwin. I'm a research scientist from Cesar Australia, and today I'm going to talk about the recent emergence of insecticide resistance in the blue-green aphid. Now, blue-green aphids, that you may or may not have come across, are widespread crop pest, primarily targeting lucerne and pasture seeds and lucerne forage crops, and also several types of grain legumes. They cause damage to these crops by firstly directly feeding, so removing nutrients, but also, and often more significantly, via transmitting plant viruses, so particularly a number of plant mosaic viruses. Now, as with most aphids, blue-green aphid typically most abundant in spring and most problematic in spring, but we know they can also pop up and cause issues in other parts of the year, particularly autumn and winter months. Now, historically, blue-green aphid hasn't necessarily been a high priority pest, and this is largely because growers and agronomists have been able to confidently and economically control blue-green aphid by using two groups of insecticides, and they are organophosphates, group 1B insecticides, and carbamates, group 1A insecticides. And for most crop types, these two groups of insecticide are the only registered chemical options for use on blue-green aphid. Now, however, in recent times, in the last 3 years or so, we at Cesar Australia receive reports of growers and agronomists that have gone out and sprayed their crops using organophosphates or carbamates, as they've done for many years gone by, except this time, these insecticides provided little to no control of blue-green aphid. Now, in this table below, we provide a few examples of the approximate location and approximate date of these control failures. Now, this table is by no means an exhaustive list of control failures. This is a table that fits on a slide without the font getting too small. But one of the main things to take away from this table is that we've had cases of insecticide resistance being reported through a reasonably wide geographic range throughout New South Wales and South Australia, so it's not an isolated area. And we've also had insecticide resistance reported across multiple time points from 2019 to essentially this present year. So it's not an isolated period where this is happening. And the other thing to take from this is that we've had reports of control failure to both types of organophosphates and carbamate chemicals. So chlorpyrifos and omethoate are the primary types of organophosphate used to control blue-green aphid, and pyrimecarb is primarily the major carbamate used to control blue-green aphid. And for both of these, well, for all three of these chemicals, we've received multiple reports of control failure. So unfortunately, these reports suggested to us that blue-green aphid had evolved resistance to these chemicals that have been registered and relied upon for use against blue-green aphid. However, we also know that control failures can occur to a number of other reasons. There can be poor application issues, there can be environmental issues, there can be issues with batches of chemicals, for instance. So before we essentially at Cesar Australia go out and make recommendations that people should change their practices, we want to be really sure if resistance has occurred in these populations, and if so, what magnitude are we dealing with? Is it a small shift, or where chemicals might still have some efficiency, or is it a complete loss of sensitivity, in which case these chemicals, they're essentially useless, at least against this species. So what we did as one of our first steps was to go out and collect multiple populations of blue-green aphid where resistance has been reported and test them against these registered chemicals under controlled laboratory conditions. So we did this for organophosphates, for both omethoate and chlorpyrifos, and also against carbamates. And this was done for pyrimecarb. So to do this, we go these laboratory studies to measure the levels of resistance. We went out, well, we actually got aphids posted to us in from growers and agronomists that experience control failures. It's one of the handy things with aphids. They can survive a journey in the mail. And once we received these aphids, we removed their parasitoids and viruses, and then we bred them up under laboratory conditions. And we did this for 3 populations. We did this from 1 population in New South Wales, Temora, and 2 populations from South Australia, Keith and Lapa. Now we tested the level of resistance of these 3 recently collected field populations against a 4th population, which we sort of term our susceptible population. Now this is a population which we have maintained in the lab for a number of years, and we know due to past work we've done with it and also understanding the genetics of this strain, that this susceptible population lacks resistance to any of these insecticides. So essentially it provides us a reference or a yardstick to compare these 3 recently collected populations to test, firstly, have they evolved resistance, and second, to what magnitude have they evolved resistance. So to do this experiment, what simply we do, once we've bred up enough aphids in the lab, we then expose them to several doses of insecticide in a controlled petri dish environment. We then follow these aphids several subsequent days to score their mortality. And then we essentially do a bunch of data analysis at the end to determine if any differences occur between populations. Are these differences statistically meaningful, or are they just noise? So what do we find? So I've talked long enough without actually showing any results. So the first results I want to show you here are for omethoate. So this is a type of organophosphate, which is again commonly used for blue-green aphid. So I'll just walk you through what this graph is showing before, I guess, discussing what it means. Now, on this horizontal axis, we have each of our 4 aphid populations that we're testing, and they're each also shown with a different color. And on this vertical axis, we have the LC50 values for omethoate. Now, LC50 is just a fancy way of saying the concentration where half the aphids died, 50% of the aphids died. And it's just a standard metric that we use to be able to quantify the level of resistance. But essentially all you have to know from this is that the higher LC50 value, then the higher level of resistance. And the little dot just shows the average value and the bars just show the variance. So if there's more noise in certain populations than others. Now, what jumps out pretty quickly from this figure is that all 3 of our field populations, Tamora, Keith, and Laffer, all had a higher resistance, a higher LC50 value to omethoate than our susceptible population. And when we quantified this level of resistance, it sort of came to a 9 to 23-fold resistance, which is sort of a bit of a medium level of resistance as far as things go. Now, this suggested that unfortunately, that these populations had all evolved resistance to omethoate. Our next step was going to test, okay, so we had one type of organophosphate where resistance evolved to. We wanted to see, has resistance just to the specific product, omethoate, or is it to the whole group of organophosphates? And so we tested a second type of organophosphate, which is commonly used for blue-green aphid, and that's chlorpyrifos. And what we found was a very similar story. So this figure essentially the same as our previous on the slide, but on the vertical axis, we just have chlorpyrifos instead of omethoate there. So we found that although one of our populations seemed to have a lower resistance to chlorpyrifos, Laffer, which is a bit of a surprise, our other 2 populations, so Keith from South Australia and Tamora from New South Wales, both showed a significant level of resistance, which was about a 30 to 40-fold shift in resistance compared to the susceptible population. So again, these results are suggesting to us that blue-green aphid has not only evolved resistance to one type of organophosphate, but seems likely to evolve resistance to the entire group. Now, again, this result isn't good news, but it's also what we anticipated based on what we're hearing in the field. So these populations are all populations where growers and agronomists have reported failures for these chemicals in the field. So at this point, we had reasonably strong evidence that blue-green aphid had evolved resistance to organophosphates. When we wanted to then go and test the second type of chemical, which is registered against them, at least for most crops, and that is carbamates. So we specifically tested pyramecarb. And what we found here is, again, unfortunately, a very similar story. We found all 3 of our field populations showed a higher level of resistance compared to our susceptible population. And again, when we quantify this, we find that it's slightly less, but still significant and still worrying enough. We found sort of a 6 to 14-fold shift in resistance to pyramecarb in all of these populations, which suggests to us that resistance has also evolved to carbamates. So what do all these results mean? So I'll walk through some conclusions of this study before going on to some of our short-term recommendations of what we can do next. So unfortunately, our research shows or strongly suggests that all field populations have evolved resistance to organophosphates and carbamates. And this is a particularly worry because in most crop types, these are the only 2 chemicals that are currently registered for use against blue-green aphid. So even though here we only tested 3 populations. We suspect, we don't know, but we suspect that this resistance has spread reasonably widely throughout southeastern Australia. Now, I'm going to come back to this point later because this is specifically where it's spread and whether it's the same strain, something we want to explore further. Now, the last thing I want to touch on is this conclusion is that our research suggests that this resistance is a heritable basis. So heritability just means it's basically stably passed from parent to offspring. And we suspect, well, we strongly suspect this because these populations were maintained in the lab for a number of generations without showing a shift in resistance. Now, the importance of heritability essentially is that heritable variation is essentially the fuel for evolution to happen. And so this point of heritability is going to be really important when we think about what management approaches to do. Because if we mismanage these resistant populations with this heritable basis, it might mean we push them even further and have even a bigger problem on our hands. So I'll touch on this a bit further as we go through our chemical recommendations for chemical control. So we recommend, where practical, avoid spraying blue-green aphid with either organophosphates and carbamates. And this comes back to this point in heritability, where essentially, if we continue to spray these populations with these chemicals, we're going to continue to put selection pressure on them, which means that although now we have sort of a moderate, medium level of resistance, we're going to push it to be even further due to this heritability. If these chemicals must be used, if you're desperate, then don't cut. We suggest that don't cut rates, as using these sort of lower rates of various chemicals will most likely allow this resistant strain to spread more. But also recommends having a plan B, because these chemicals simply just might not actually work. In some crops, so in some pulses, there is other registered options, and that is sulfoxaphor, transform. And we've had no reports of resistance from this chemical, and it acts in a very different biochemical way than organophosphates or carbamates. So for the time being, where crop types where sulfoxaphor is registered, we would suggest that this is the best chemical option. In some lucerne crops, there has been an emergency permit approved for specific timeframes, and that's flumiconid. And again, that would be where it's covered by this emergency permit by the APMEA. We suggest that this would be the best option. So outside of these chemical controls, and particularly for crops types where only organophosphates and carbamates are registered, there's other alternative ways we can control blue-green aphid. And the first one to touch on is biological control. So we know that many natural enemies, so hoverflies, lacewings, ladybirds, and parasitoid wasps, can be effective at suppressing blue-green aphid. And we've had very positive anecdotal reports from growers over the last couple of years that experienced these initial control failures from of blue-green aphid, that then effectively manage the natural enemies to not remove blue-green aphid entirely, but at least maintain blue-green aphid below damaging levels. So a key part of managing these natural enemies is selecting chemicals less likely to harm them. And to help you with this, we've at Cesar Australia have developed a toxicity guide of natural enemies, primarily targeted to the grains industry, but number of these natural enemies are known to attack blue-green aphid in multiple crop types. So we'd suggest, when you're selecting chemicals, not only for blue-green aphid but for other pests, just being mindful of what chemicals you use, and if these chemicals are going to harm known natural enemies of blue-green aphid. Other recommendations covering culture options. So we know, at least for some crop types, particularly sort of lucerne and I think some clovers, there are particular cultivars that are more resistant to aphid feeding damage than others, and we would suggest, where possible, making use of these cultivars. The other thing to be mindful is that blue-green aphid are typically most abundant in spring and early autumn, but between these periods, they often will move to alternative hosts between growing seasons. And these are typically type of weeds, hypermedic weeds. So being able to control these alternative hosts in between growing seasons or remove them can really help at least delay the arrival of blue-green aphid into your crops. The other recommendations we have, so with managing any species of pests, but particularly with aphids, there are multiple species that can essentially that co-occur with blue-green aphid and attack the same crops as blue-green aphid, and can sometimes be difficult to identify. So we would strongly suggest making use of this GRDC back pocket guide on crop aphids that we developed to sort of help you if you do come across this species, particularly being able to identify the difference between a blue-green aphid and a pea aphid can be quite difficult, but making use of this resource can really help your decision making from that point on. And on this, Cesar Australia is also running a bunch of insecticide, insect identification courses over the next 12 months. So just being abreast of our socials when these courses are happening, if they're of interest to you. So here are some of our resources. So here to help you post this talk. So we've written up a pest note on our website, which essentially summarizes some of the parts of this talk and also management practices that can be used for blue-green aphid. This is the GRDC back pocket guide, Cesar Australia's toxicity guide for natural enemies, and if you have any further questions, or if you suspect you have resistance popping up in your area, please get in contact with me. And I'll leave this slide, I guess, up for a second if you want to screenshot it or photo. Yeah. But before I go, and a couple last things to sort of return to this first slide here is that, as I say, this is the first time blue-green aphid has evolved resistance anywhere in the world. And this is despite, we know that blue-green aphid is a huge pest all throughout the USA and all throughout North America, South America, parts of Asia, definitely parts of Southern Africa, Europe. But despite this, resistance hasn't popped up there, at least there's no reports of it popping up there. It's happened here in Australia. And what does that mean? It's not about finger pointing to any particular person or industry of why this might have happened, but it's something that we really need to be mindful of going forward. Because if there are practices that are currently in place right now, which drove blue-green aphid to evolve resistance here, but nowhere else, that might also mean we might get other species of pests that evolve resistance here. Or we might see blue-green aphid evolve resistance to other chemicals that we might get registered to them in the future. So this is one of the take-home messages, is more broadly, we do have to start thinking about why this has happened. And as part of this broader thinking, Cesar Australia is continuing our research on insecticide resistance and blue-green aphid. One of our key next steps in doing this is really mapping more detail where and what crop types these resistant strains of blue-green aphid are spread to. And we really need your help to essentially do this to the best of our ability. So currently we have information from 3 populations, but we want to get as much geographic spread and crop variation as possible. So if you see blue-green aphid in your area, particularly from pasture seed crops and grain crops, usually pulse crops we're interested in, and clover seed crops. We would love you to get in contact with us. If you hear anywhere down the road your neighbours experience issues with insecticide resistance of blue-green aphid, please, send me an email, give me a phone call, and we would love to get these populations in the lab. Because at the end of the day, the more populations we can test, the more detailed recommendations we can put out. We can give more specific recommendations to certain regions and also growers specific crop types. So that's essentially our end goal, at least for our next, well, not end goal, but our next goal. And you can get in contact with me through either social medias or direct email. And I think the contacts are also on the Cesar Australia webpage. I think that's it. I'll leave it there. Before I end, of course, I'd just like to acknowledge the main funders of this work, which was primarily funded by AgriFutures. Our initial steps in this space were funded by GRDC. We also just a great thanks to all the growers and agronomists that posted us in blue-green aphid and also spent time on the phone chatting about how and where they've had issues with this pest. Again, if you have any emails or any questions, shoot me an email or give me a call. And thanks.